There's the A for the day. I will give you an A. An A for today won't make you much tomorrow, but at least today you can feel like, you know, what did you do today? You can say, I made an A. I made an A of myself, I think you can say. That might not be a good thing to say. How's everybody doing? Studying's coming along? Okay. Things making sense?
Ish.
No. Ish. Okay, so hopefully things are making good sense, and what you need is review, but there's all kinds of ways of getting at this material. So the way this will work is I will go as long as you ask questions, unless you go about 3 days, in which case I may cut it short before then. But it's driven by your questions. I have no presentation per se that I will do, and that's so that anybody who doesn't want to come to the review session doesn't miss out on anything. So anybody who wants to ask questions can, whether they are coming to my office or they're coming to review sessions. So, and I will work through those. So that's kind of where we start. I will get this posted on the web as soon as I can. I'll start the video rendering as soon as this is over, and I would expect it should be posted on YouTube sometime this evening. So we'll see about that. All right. So, what are your questions? Yes.
Hi. For HPLC, what in the world is stopping the hydrophobic things just like staying in the column?
For HPLC, what is stopping the hydrophobic things from just sticking to the column? Okay? Okay. Any kind of interaction between a protein and something else that does not involve a covalent bond is not a permanent thing. It's an association thing. So the analogy I would give to that is that there are certain people that you are fond of. I've used your significant other, and other people that you're not so fond of. And so if you were walking through a crowd of your significant others, it would take you longer to get through that crowd than if you were walking through a crowd where everybody had BO. Right? All right? So, but you will still get through that crowd of significant others. Let's face it, significant others don't always last forever. Right? So you may be attracted to, but you're not bound to. And that's true of any non-covalent interaction that we talk about. So it's even true of the ionic ones that are there. Those things, if we get them long enough, will come off of the column if they're charged plus attracted to a minus. They just won't come off as fast as something else will. Does that make sense? So when I say they're stuck to the column, it's sort of a metaphor. It's not a real thing. They're not there forever. Yes, sir?
Is the practice exam that you posted pretty representative of the format of the actual exam?
The practice exam I posted is exactly the format of the exam. That is, you will see the same. The only thing I would say is different is the point distribution. I can't remember. In fact, I'm not sure that the practice exam has a point distribution. I can tell you that the final exam has three the first exam, I'm already thinking, you can see I'm ready for the term to be over. Actually, that's not true. I like teaching this class. The final exam has... The first exam has 3 sections. It has the same first section as the practice exam, which is that it has a short answer section. It has a problem-solving section, and it has a longer answer section. And the distribution of points across those is that there's 40 points in the short answers, there's 30 points in the problem-solving, and there's 30 points in the longer answering section. I can tell you that there are, in the problem-solving section, that there are 2 problems, and in the longer answer section, there are also 2 problems. And there are 2 extra credit questions. You guys sang beautifully yesterday. Yes, sir.
When we talk about hemoglobin, there's a heme, right? When the heme bonds with oxygen.
Yes.
And the histidine will help to stabilize the oxygen with the
Histidine
so
does.
I want to know, is the histidine which connect to the heme directly to help stabilize the oxygen, or the other part of the polypeptide bonds?
I'm not sure I understand your question.
I mean,
So the histidine relative to the heme. What's your question?
yeah, I mean, when the histidine help to stabilize the oxygen
Yes.
with heme, I
Okay. All right. So his question has to do. I'm repeating the question so because people on tape on the video can't always hear. So his question has to do with the histidine involved with heme, and there actually are 2 histidines involved with heme. I haven't talked about the second one, and that's the one that you're asking about. He's asking about the one that stabilizes the oxygen. Okay. And the reason I haven't talked about that histidine is it confuses people. There's a histidine below and there's a histidine above. And the histidine above plays a very important role. The histidine above does not touch the heme at all, but it does interact with the oxygen. You're not responsible for that one because I have not talked about it. But since you asked the question, I will tell you an interesting thing about it, and that is that the histidine above actually favors the binding of oxygen over carbon monoxide. Okay? Carbon monoxide can bind, and in fact it does bind to the iron of heme. But with that histidine up there, it makes carbon monoxide slightly less favored. It still can bind, and it still causes major problems. It's one of 2 reasons that carbon monoxide is poisonous to you. But that's what's up with that. But I have not talked about that second histidine on purpose.
Another one.
Yes?
When the environment, when the pH of the environment drops, so there are going to be more protons, right? And so they will connect to the histidine in the hemoglobin. So I mean, this histidine is still the bulk of the histidine.
Okay. So his question has to do with when the pH is low, there are more protons. And I said that the protons bind to hemoglobin and favor the release of oxygen. And the question is, does it bind to that histidine? And the answer is no. It can bind to other histidines in the molecule. It can also bind to amine groups in the molecule.
When the proton binds the histidine, it's going to be positive charge, and
If protons bind to histidine, yes, you'll have a positive charge.
attractive some other amino acid and then change the structure into the histidine.
So his question again has to do with binding of protons to various groups in hemoglobin, and the answer is that yes, the binding of protons to various groups inside of hemoglobin does, or actually is the cause, or one of the causes of that shape change. You're making the T state, okay? So that T state favors the release of oxygen. So yes, that does happen. And again, it comes back to this theme I've talked about all term about slight changes in charge, slight changes in molecules bound to the hemoglobin have very slight shape changes, and those shape changes are really what's converting hemoglobin from the R state to the T state or from the T state to the R state. Okay, so those are all very important considerations. Yes. Yes, sir.
Just had a question about the exam, practice exam wording on question 2 said, name and describe the structure of proteins that gives rise to alpha helices, folding, and subunit interactions. Are you just talking about the primary, secondary, tertiary, quaternary, or are you looking for something different?
Okay, so that probably, let me just read the question so that people out in TV world will hear it. This question I get all the time, okay? And I don't recommend studying old exams, all right? Old exams, I talk about things different from year to year, and as a consequence, you can spend a lot of time, and this question is one of them, trying to figure out what in the heck he's talking about because he hasn't talked about that this year, okay? It's in the wording, okay? So the question is, name and describe the structure of proteins that gives rise to alpha helices, folding, and subunit interactions. Well, if you think about it, what's alpha helices? That's secondary structure. What's folding? That's tertiary structure. What's subunit interactions? That's quaternary structure. Now, if you think about that, what did I say was responsible for all of the structures of a protein? The primary structure, right? Okay. But again, you're going to see things differ from year to year. Don't study old exams, okay? Use the practice as understanding my format. You'll spend much better time studying the material than you will studying practice exams. Yes?
Do we have to know how, so I understand how to do a titration curve
Good.
for one
You get an A
amino
for the day, too.
acid? For one amino acid, but will we have to know how to do it for a polypeptide?
There's no difference in doing one for a polypeptide than there is, I can't say if you will or you won't, but there's no difference in doing one for a polypeptide than there is for an amino acid because all you have are pKa's. That's all you're plotting by. So if I gave you 12 pKa's, you would say, okay, here's what it looks like because each flattening place is corresponding to a pKa. So it doesn't matter if it's in one molecule or 10 molecules. There's no difference. You see what I'm saying?
Yeah, I tried to find the PI for one for a polypeptide, and I thought I was right, and I got it wrong, and I don't know why.
Well, would you like me to do a PI?
That would be great.
That would be great.
For a polypeptide.
for a polypeptide. Okay, so I'll make up one. How about that? So let's. I'm going to raise the screen. And I'll have this available if I need it, but for the moment, I'm going to turn that off. Okay. Now, her question, maybe I should turn the light on. There we go. Her question has to do with how do I find PI for a polypeptide. So let's make one up. Let's say I've got leucine, aspartic acid, lysine, and isoleucine. Okay? And the pKa values I gave you were that alpha amino groups have 9.5, alpha carboxyl groups have, let's say, 2.2. R group carboxyls have a pKa of 4.4, and R group aminos have a pKa of, let's say, 11.5, just for the sake of argument, okay? So if I have those, then what would the PI for that guy look like? Well, I really like drawing a titration plot for anything I'm working with because the titration plot really helps me to understand where the charges of these groups are. So what do I have here? I have an NH3. I have over here a COOH, and I'm putting the protons on at the moment. This is with an R, and an H3. And I have a COOH. So I've got the protons all on there. If I plot pH versus OH added, what's this guy going to look like? Well, when I start at a low pH, all the protons are on. I get to the first buffering region. It flattens out. It rises to the second buffering region, rises to the third, rises to the fourth, and finally gives up. And I know I've got 4 because I have 1, 2, 3, 4 groups. Okay? I could have put this onto one molecule. So it didn't matter if it was 4 molecules or 100 molecules. All right? So now I start filling in my stuff. So here's the first pKa, which I said was 2.2. Here's the second pKa, which was 4.4. Here was the third pKa, which corresponded to 9.5. And here was the fourth pKa that corresponded to 11.5. All right? Now, the next thing I would want to do on my graph is look at this and say, well, what's the charge at each place that I can tell the charge? Well, there's only certain places I can tell the charge, and that's where the protons are all on or all off individual groups. Way down here, pH is 0. So protons are going to be on all the groups because the pH is below the pKa of all those groups. So I put the protons on. I put the proton on here, I've got a plus. I put the proton on here, I've got a 0. Proton on here, I've got a plus. Proton on here, I've got a 0. So this guy has an overall charge of +2. Okay, I can't tell here because I got half of them on, half of them off that first group, that first carboxyl. So I can't tell charge here, but I can tell charge here because I have this first proton that's come off. This pH is roughly, let's say, 3.3. And at that pH, I can look at that and say, well, pH of 3.3 is above this one by more than 1 unit, so this proton is off. That's the alpha carboxyl. So the alpha carboxyl has proton off. That's got a negative charge, so I've got a -1. And then this pH is below the pKa of all of these, so their protons must all be on. So I've got on, off. And so you see my charge now is +2. It's gone to +1. Well, not surprisingly, every time I go through one of these, I'm taking off one more proton. I take a proton off here, I take a proton off here, I take a proton off here, and I take a proton off here. Each step is a proton, right? Well, if I want to calculate the PI, it's got to be the average of the pKa's on either side of the place where the charge is approximately 0, which is here. So the PI for this guy would be 4.4 plus 9.5 divided by 2. Make sense?
Yep.
Clear as mud. Okay, you guys have done these in recitations. You've seen these. Does that make sense? Do you have questions on that? I want to see everybody get this right on the exam. Okay? That would be my dream. Everybody get them all right on the exam. Yes, ma'am?
Just to double check, that's an acceptable answer, right? We don't actually add it.
I said you don't have to do any calculations whatsoever. All right? No calculations whatsoever. Yes, ma'am?
And will you let us know which R groups are on which?
No, you need to know which R groups are on which amino acids, right? Which category they fit into. This guy is this guy is the one that has the carboxyls, the acids, right? This guy is the one that's in the R groups with the amines. I'll give you the full names. I'm just abbreviating them here, but I'll write lysine. I'll write aspartic acid. You don't need to know the three-letter abbreviations. But no, you're responsible for knowing. That's why I say you've got to know what groups they fit into. And as a consequence of that, you've got to know which ones ionize, right? So definitely you need to know that. Yes, sir.
Given in a problem like this, would we have a value for pKa's listed?
Always, yeah, sure. Yeah. If you look on the practice sheet, you'll see I already give you pKa's on there, I think. If not, let me see what it says. What does it say? Are they? They're not on there. But usually I'll put them on the bottom of the first. It'll be something like this. It'll say alpha amino, you may assume this. Alpha carboxyl, you may assume this. Okay? And that's what you'll see on there. But yes, I give them to you. Yeah. But I'm not going to tell you, lysine has an R group that's got an amine on it. No. You need to know that's an ionizable group that's on there. And then when I say R group amine, you know this, right? So that's why. And usually this is the way I'll summarize it in that table. Yes?
So which amino acids do you want us to know all 20 of them?
You need to know all 20 amino acids. You need to know the names of all 20. You need to know what groups they fit in. And if you have any questions about what you're responsible for in those amino acids, go back to the lecture that I talked about this. I said explicitly what you needed to know about those. Yes, sir?
Show us how to do a reverse application problem on the sample.
On the sample exam? I don't know what the first problem is. Okay. So there's a story about this problem, and I've told several of you this story, but this first problem that's on here was sort of a nightmare problem for my class because I wrote the problem without realizing at the time how complicated the problem was. And it was just an oversight. Sometimes you write problems, you don't really realize that. And so this was the nightmare problem. And everybody always wants to know about how you work this problem. All right. When I put this problem out on an exam, there were 300 people in the class, and I think 3 people got the problem. Okay. Well, I felt horrible. They felt horrible. They weren't happy with me, needless to say. But the curve was low, and so grades aren't absolute, so balanced out. But on this exam where I asked this problem, I had the lowest average I ever had in a class. The average was 47, okay? Only time I've ever had an average below 50. I felt really horrible. And believe me, I feel as bad as you do when those are low. Because I think, if you're not getting a certain percentage of what I talk about, then that's probably my problem. Well, anyway, this problem was there. So I actually, one of my songs that I wrote, if you look at my songs, you'll see there's a song called Biochemistry Pi. And it's about that class. And there's a line in there about the average being 47, and that's what it's referring to. So anyway, okay, but I'll work this problem for you. So this is a complicated problem. You have 500 mils, okay, of a buffer of a concentration 0.4 molar, okay? pH is 6. All right, then add 0.05 moles of NaOH with no volume change. And there's salt over acid equals 2. Now, one of the reasons, here you go, thank you much. One of the reasons people had such problems with this was they just looked for numbers and they looked for equations and they plugged them in. And they said, well, I know from Henderson-Hasselbalch that pH equals pKa plus the log of salt over acid. Okay? And look, I've got a pH. Okay, that's good. That's 6. And I've got salt over acid. Okay? And I've got pKa. So I only have one unknown. So pKa, therefore, equals 6 minus the log of 2. Probably 95% of the people in the class wrote that because they didn't read the problem. Okay? What did the problem say? The pH was 6, and then I did something to it. Okay? It's not 6 when it's 2 to 1. It's not 6 at that time. So here's one solution. Here's a different solution. They're trying to work the same equation for them. Doesn't work. Oh. All right. So what do I have to get rid of this thing? What do I have to do? Well, what I need to do is find out basically what the S over A was when the pH was 6. I have to go backwards. I have to go backwards in this problem. All right? So let's think about this. How do I work this problem? Well, I've got a buffer. When I say the word buffer, the first thing that should pop into your mind is salt plus acid equals buffer. Salt plus acid equals buffer. Okay? How much buffer do I have? 0.4 moles per liter times 0.5 liters equals 0.2 moles of buffer. So therefore, salt plus acid equals 0.2. Right? Salt plus acid equals 0.2. Okay. When the ratio is 2 to 1, if we do the math, what you'll see is this turns out to be 0.13 of salt and 0.06 of acid. 0.66 basically. Okay. That's what those two are. When it's 2 to 1. But it's not 2 to 1 when pH is 6. What were the amounts that were there when the pH was 6? Well, what made it be 2 to 1, guys? The addition of the sodium hydroxide. What if I go backwards and subtract the sodium hydroxide? When I add sodium hydroxide, I convert acid into salt, right? When I subtract sodium hydroxide, I've got to be converting salt into acid. So the 0.05, I'm going to convert salt into acid. I'm going to lose 0.05, get whatever that is, 0.128, and I add 0.05, and I get what? 0.116, right? Something like that. No, I'm sorry. I can't. I don't know why it's, okay, that's fine. But that's basically what I get with those two, right? That's what it was when I added, before I added the sodium hydroxide. I now know what it was when it was pH 6. Right? When it was pH 6, this was salt, this was acid. All right. Kabingo, right? I can now go through. Here, I knew I did that wrong. It's 0.083. That's what it is, right? Okay, now that makes sense. I got to learn to do math. This is what it was when I had the pH 6. So now I can say pH, which is 6, equals the pKa plus the log of what it was originally, 0.083, and the acid was 0.116. I can now say that pKa equals 6 minus the log 0.083 all over 0.116. That's how the problems work. It's a tough problem. You had to recognize you couldn't just plug and chug because you were comparing apples and oranges, and then you had to work backwards. And so as a consequence of that, 3 people out of 300 got it right. So hopefully I haven't made up a horrible one for you on this exam. Yes?
So then for when you remove the NaOH, why do you remove 0.05 instead of salt and add 0.05?
Well, if I add sodium hydroxide to it, normally what I would do is I'd convert acid into salt. I would subtract from the acid and add to the salt, right? So if I take it backwards, I've got to basically reverse everything I did. I've got to remove from the salt and put it back on the acid. Right? Make sense? Other questions about that? When I add sodium hydroxide, I always lose one, gain the other. If I add HCl, I lose one and gain the other, right? because all I can do is convert one to the other with protons or hydroxides. The only way I can change the amount of one without the other is if I add one without the other. Let's say I added sodium acetate to that solution. I'm not adding any protons. I'm not adding anything else. If I added sodium acetate to that solution, I would change only the salt. I would not change the acid. Right? That's what happens when I manipulate those. Okay? So recognizing what happens with protons, recognizing what happens with hydroxides is essential for understanding what's happening to the relative amounts of salt and acid that's in that solution. I'll tell you, this problem is hard enough. I had to show the TAs had to work it. So it was a mistake on my part. It shouldn't have been there. Yeah?
If we're given a sequence of amino acids, how do you tell what side the carboxyl group or the amine group would be?
What side it's on?
Yeah, if you were going to treat it with an enzyme.
Oh, okay. Well, the convention in general is that when I write the sequence, the amino end is on the left and the carboxyl end is on the right.
Oh, okay.
That's the convention. Okay? So that will always help orient you when you're making those cuts. And her question was, maybe I should repeat that. Her question was, when you have a sequence of amino acids, how do you know which side is the carboxyl end and which side is the amine end? And so the answer is that the convention is you write it so that the amine is on the left and the carboxyl is on the right. Yes?
So when you add sodium hydroxide, you gain salt.
If you add it to a buffer,
Why are you losing salt? Why is it
I'm going backwards. I'm taking away sodium hydroxide. I'm going back before I put the sodium hydroxide in. I have to reverse what I did, right? So I'm reversing what I did. Yes?
It appears like this problem, in order to get the answer for PK, you have to have conversations.
No? Oh, you mean to get the number?
Yeah.
That's the answer right there.
Oh, okay, you don't.
There's a difference between answer and number, right? All right. That is the answer. Period. Okay. I'm freeing you of a calculator, folks. I can't tell you the number of times I've seen students do the whole thing on a calculator, and they come up with a number that makes no sense whatsoever, and they probably did it right, except for they missed a decimal point in the very last step. We can't see their work. They get a 0. I'm making you show me what you do if you want extra credit. That's the way you get extra credit. In fact, I'll emphasize that. On this exam, the more organized you are and the more you label things, the more the TA can understand your logic and the more the TA can give you extra credit. And if you have some scribble scratch on there and there's nothing labeled, if it's wrong, it's wrong. When you get to the end, it's wrong, it's wrong. So label things. Learn to do that. It will also help you keep things organized in your heads. Neil?
So besides fitting into the donut hole, when 2,3-BPG, when it enters, how does it input the T state?
So his question is, hemoglobin has a donut, and 2,3-BPG fits in the donut hole, so how does it convert it to the T state? The answer is, just like protons cause small changes inside of proteins, so too does binding of molecules to hemoglobin change its structure. It turns out that the structure of the 2,3-BPG is such that it fits perfectly, and just like a wrench, it turns it to T state. Just like if oxygen binds, it turns it to R state. So these very tiny changes are what ultimately cause it. I'm sorry?
There's no specific linkage or anything between the amino acids and hemoglobin?
His question is there any specific linkage between the amino acids and hemoglobin? The answer is absolutely. Just because I haven't told them to doesn't mean that they're not there. But 2,3-BPG is not going to bind unless it makes interactions. Interactions are essential for that, right? So yes, interactions are the root of every single thing that's happening in the structural changes of a protein. That's absolute. Otherwise, it's magically doing it. And we don't like magic in science. Was there a question over here? You have a question? Okay. Yes, back in the back.
Can you just give a simple explanation of how carbon dioxide affects hemoglobin making for oxygen?
Can I give an example where carbon dioxide affects hemoglobin's affinity for oxygen? Okay. So there was the graph that I showed, and the graph basically showed that it was used to illustrate part of the Bohr effect, and the Bohr effect includes both the protons and the carbon dioxide. So I'll show you the graph, and then I'll try to tell you something in words. So if we look at the graph where we have percent saturation of hemoglobin versus oxygen concentration, okay, what we see is this sigmoidal plot that looks something like that. And this sigmoidal plot is indicative of cooperativity. When we do a measurement like this, we of course do it in a buffer. We do it in a specific pH. And in this case, the pH they did it was the pH of human blood, which was 7.2 or 7.4. And then Dr. Bohr went and wisely said, well, what happens if the pH is slightly lower? Because when I have respiring tissue, the pH is lower. And it turns out that that's what the plot looks like at 7.2, indicating that I have a smaller amount of hemoglobin saturated with oxygen at any given oxygen concentration because when I have this lower pH. That means that when hemoglobin encounters rapidly respiring tissue where the proton concentration is slightly higher and the pH is slightly lower, that more oxygen will be given up, and that makes very important sense for respiring tissue. The third component of that is if we do the same experiment at pH 7.2, but we add carbon dioxide, then what we discover is that the binding curve is even lower, and there's even more oxygen being given up. Now, that wasn't, I think, what your question was, but your question was, what does this mean with respect to the binding of carbon dioxide to hemoglobin? As you saw on the picture that I showed you, carbon dioxide makes a covalent bond with carbamate ions on the, with the amine groups on side chains on a hemoglobin, and that makes what's called a carbamate molecule. That carbamate molecule is on there basically until the carbamate gets back to the lungs. The addition of that carbon dioxide changes the, again, shape, charge of hemoglobin such that we are twisting hemoglobin a bit and favoring the release of more oxygen. Okay? Now, one of the things I didn't talk about this year, but I will mention it to you because the question is probably in your head, especially after I said about covalent bonds. Boy, those are really stuck on there. It turns out that covalent bond to the amine group in hemoglobin is not very strong. And it can, in fact, reasonably be broken. And what happens in the case of that bond is that when hemoglobin gets back to the oxygen, it gets back to the lungs, and there's abundance of oxygen, that oxygen literally forces itself onto hemoglobin and causes a change that favors the breaking of that bond and the release of carbon dioxide. So it's the high concentration of oxygen in the blood that not only favors the conversion to the R state, but literally the release of the carbon dioxide. And that's why you exhale carbon dioxide when hemoglobin gets back to your lungs. It's a remarkable thing. I mean, it's absolutely remarkable. Does that answer your question?
Yeah. But so does that mean that CO2, like I know they function differently, but does it have the same effect as CO2?
Does CO2 have the same effect as 2,3-BPG? The answer is effectively yes, it does. Okay? Effectively, yes, it does. The difference being that 2,3-BPG ideally doesn't make it back to the lungs. Because 2,3-BPG, you want that to come off while it's in your bloodstream so that you're not stuck in the T state when you get back there. All right? If you're stuck in the T state when you get back there because of 2,3-BPG, then you've got problems. Okay? And that's why the smokers have a more difficult time breathing because they've got more 2,3-BPG in their bloodstream, and it's more likely their hemoglobin will get back there with 2,3-BPG. Okay? Question over here?
Yeah, you answered it.
I read your mind. I win the prize. Yes, ma'am.
So carbon dioxide's not forcing it to be in a T state like 2,3-BPG is?
Well, okay, so her question is, it's not forcing it to be in the T state. It's not forcing it as such to be there because, as I said, it's relatively easy to break that bond and release it. It is favoring the T state, yes, okay? But It's really set up. Hemoglobin is set up very well to let go of carbon dioxide. It's not set up so well to let go of 2,3-BPG. Okay? And it's a concentration thing. If 2,3-BPG is high as it is in smokers, it's just much more likely you're going to have more of that tied up. That's not the case with carbon dioxide. Yes, ma'am?
Sorry if you already answered this, but what prevents 2,3-BPG from returning with the hemoglobin to the lungs?
The question is, what prevents 2,3-BPG from returning with hemoglobin to the lungs? Nothing. But it's less likely. These are all likelihood things, folks. Keep in mind, we're talking about billions of these molecules. This is a likelihood thing, all right? The likelihood is, do you have any hemoglobin that makes it back to the lungs with 2,3-BPG? Absolutely. Every one of us has hemoglobin that makes it back to the lungs with 2,3-BPG in it, all right? Who's going to have the most? Smokers, right? So it's all a matter of degree. All these, remember I said when we talk about enzymes, and I said we think of enzymes as being on or off, but it's really more like we turn the volume up or we turn the volume down, right? The same thing is the case with the binding of 2,3-BPG. We're going to have some of it in our hemoglobin in our lungs. Yes, sir?
So then is the majority of it reuptaken by cells or broken down and utilized in solution on the way back?
Okay. So is the majority of 2,3-BPG taken up and metabolized by cells? And the answer is yes, it is in the non-smoker. Yes, it is in the non-smoker. In the smoker, that's not the case so much. And that won't make sense until we talk about glycolysis. It turns out that 2,3-BPG is a byproduct of sugar metabolism. And so cells that are rapidly metabolizing produce more 2,3-BPG. And in smokers, they're rapidly metabolizing because for a couple of reasons, they have less oxygen carrying capacity. It becomes a catch-22 at a point.
Would that make it a consideration for diabetics then as well?
Would that make what a consideration?
The 2,3-BPG concentration, since it's a byproduct of glycolysis?
His question is, would 2,3-BPG be higher in diabetics than it would be in non-diabetics? And no, basically not. But again, I'll have to talk about glycolysis before you see that. Diabetics don't have more glycolysis going on than non-diabetics. Right? They have a higher glucose concentration because their cells are not taking it up properly. But that doesn't mean they have more glycolysis going on. I'm talking about more glycolysis going on in the smoker because they literally have less oxygen carrying capacity. But I've got to talk about that when I talk about glycolysis. Yes, ma'am?
I know that carbon dioxide, when dissolved in water, can create acid.
Mm-hmm.
But the carbon dioxide in that graph is just the carbon dioxide that binds to the hemoglobin and is not affected with the pH at all. Right?
Okay, so her question has to do with carbon dioxide and its movement in the bloodstream, basically. She says that carbon dioxide can be converted to carbonic acid, and that's what carbonic anhydrase actually catalyzes. That enzyme I've been talking about that's so efficient. It catalyzes the formation of carbonic acid, which ionizes to form bicarbonate. So it can travel in our bloodstream freely as bicarbonate. It can also travel bound to hemoglobin. If I add this to an aqueous solution, I will get some carbonic acid that will be made. All right? All I'm saying is I've added carbon dioxide, whether it's bound to the hemoglobin or whether it's going as bicarbonate. Now, in this case, clearly some of it's going to bind to the hemoglobin, and when it does, it's going to change hemoglobin's affinity for oxygen. But I won't tell you that, again, everything is going to be in the form of that bound to hemoglobin. Some of it's going to form bicarbonate. Okay? Good questions. What else you want to know? What's the answer to question number 3, right? Yes? Is CO2 a non-competitive inhibitor? Remember that we're not talking about an enzyme. So we talk about inhibition when we have an enzyme. But it's a good question, okay? So if I were to ask you, maybe I should think about this for next year. I won't ask you that this year, but I'll ask you to think of you guys can write questions for the exam for next year. If you were to categorize CO2 based on what you know, let's say the hemoglobin were an enzyme and this carrying of oxygen is actually catalysis. Would you call it a competitive or a non-competitive? I hear both. Okay, so a competitive is going to do what? Bind to the active site. In the case of hemoglobin, we would say the active site is the heme group. Is carbon dioxide binding to the heme group? No. Carbon monoxide is binding to the heme group. Carbon dioxide is forming that covalent bond on the amines, remember? So we would say that carbon dioxide would be a non-competitive. Carbon monoxide would be a competitive, and that's a competitive inhibitor that will kill you because it's knocking out the function of your hemoglobin. Carbon monoxide, as I said earlier, and I'll mention later in the term, can kill you actually I'll mention it next term, will kill you two ways. One, by blocking your body's ability to carry oxygen, and second, by stopping your body's ability to make energy. And that's a deadly combo. Yes, sir?
Going along with the same idea, would it better fit into a chemical modifier?
Would what?
The CO2?
Would it fit into a chemical modifier? Well, it's a transient modifier because it's changing. So I would say no, I wouldn't put it in that category either. DIPF, when I put it on there, it sticks on there. Carbon dioxide's coming on, and it's later coming off. So I would say transiently it's a chemical modifier, but it's not a permanent modifier. Okay? Good question. You guys are thinking broadly. This is very good. I've never had a class doing this so well. Everybody's going to make A's. I would be delighted. I will tell you this. There's nothing that makes me happier than seeing a high average on my exam. There is nothing. Okay? And I know that you get anxious and nervous about exams, but I probably am just as nervous and anxious about the exam before you take it because I'm worried, oh my God, did I write something stupid? Oh my God, did I make the impossible problem, right? So I'm always very just on the day of the exam. So if I'm grouchy, you know why. Yeah?
On the practice exam, there's a very specific question that involves how trypsin works.
Yep.
Would we have to have similar technologies like protease?
I don't know the problem that's on there, but I will tell you that, again, studying practice exams is not a good idea. I'm talking right now about mechanisms, and some of those questions may have come. We rearranged when we talk about things each time. So I don't know, but I would say if you haven't heard it this term, then probably I'm not going to talk about it because what I talk about in class is the highest priority. Yes?
Can you go over the Lineweaver-Burke plots?
A Lineweaver-Burke plot? Sure.
How Kn acts as Kn?
Sure, sure. Okay, so the question is, can I go over Lineweaver-Burke plots and talk about how Km and Vmax and all those things appear on those plots?
So maybe also talk about inhibition with that?
Sure,
yes.
sure. Let me just do a broad thing here. So I'm going to talk about V versus S, I'm going to talk about Lineweaver-Burke, and I'm going to overlay inhibition on top of these. Okay? Let's say we start with the V versus S. So we've got velocity versus concentration of substrate. And we have a Lineweaver-Burke plot where we have 1 over the velocity versus 1 over the substrate. Okay? Now, if I take, and you remember how I set up for you how we do the experiment? We take 20 tubes, and into those tubes we put the same amount of buffer, put the same amount of enzyme, but each tube has a different amount of substrate. Those that have the most substrate will have the most velocity because we measure velocity by the concentration of product per time. Okay? And those that have the least substrate will have the lowest velocity. So if we plot those out, we will see a hyperbolic curve that looks something like that, where this tube had the least substrate and this tube had the greatest amount of substrate. And again, that's what the plot is telling us. Had the greatest velocity as well. Well, I connect these. Okay? And I see a plot that looks something like that. Well, from this plot, I've drawn it fairly simply, so I can fairly easily show or calculate what Vmax is. And there's my Vmax for that reaction. Now, remember that the concentration of substrate it takes to get to Vmax tells us nothing, because it essentially means we have to have infinite amount of substrate to saturate the enzyme. So that doesn't tell us anything about the enzyme's affinity for its substrate. So instead of using Vmax, we use Vmax over 2, because it doesn't take an infinite amount to get there, and the relative amount it takes to get to Vmax over 2 is a good comparator between different enzymes. So if I go to Vmax over 2, and I look at the substrate concentration it takes to get that, that is the value we call Km, the Michaelis constant. Km is a measure of the affinity of the enzyme for its substrate. Enzyme that has a low Km will have high affinity for its substrate, because the Km, we remember, is a concentration. It's not Vmax over 2. It is the substrate concentration it takes to get to Vmax over 2. Don't forget that. Okay? Km is always a concentration. Well, if it takes low concentration to get to Vmax over 2, this enzyme must be grabbing it. It loves it. If it takes a high concentration to get to Vmax over 2, the enzyme is asleep for a long time until it starts getting hit in the head with enzyme when the concentration is high. It has low affinity for its substrate. Okay. Well, if I take that same information and I take the information, I take the velocity and I invert the value of the velocity, and I take the substrate concentration, I invert the value of the substrate concentration. You did a problem like that out of the book, okay? What you discover is that when you take that data and you plot it on a plot of 1 over v versus 1 over s, that what was a hyperbolic function becomes a straight line, okay? As straight as I can draw one anyway. And the reason for doing that is that we get 2 points that are very valuable to us, very easy to determine. We don't have to guess where's Vmax here. We simply have to read it off of this plot. So here are my points up there. And now I know that the y-intercept is equal to 1 over Vmax, and the x-intercept is equal to minus 1 all over Km. So those 2 values that might have had some variability over there pop up precisely over here. So the Lineweaver-Burke plot really helps us to determine these values quite readily. Well, what if we have inhibition? That was the question that was asked. I've told you that you're responsible for 2 different types of inhibition. You're responsible for understanding competitive inhibition and also for understanding non-competitive inhibition. All right? Now, I've got some different colored chalk, so I'm going to show you different things with the different colored chalk, so it'll be really high-tech here. And let's think about it. Competitive inhibition, what do we have? Well, we have a molecule that looks like the normal substrate for the enzyme, and it competes with it. It goes and it binds at the active site. It binds at the active site. It's not bound permanently, but it's bound there. And when it's bound there, it's preventing the substrate from binding. If my substrate, when I do this experiment, I take my 20 tubes, I have the same enzyme and the same buffer, but varying concentrations of substrate. You remember I said when we do the inhibitor, how much inhibitor do we put in each one? Put the same amount, right? Because we don't want to have other variables. We have the same amount of inhibitor in each one. If the ratio of inhibitor to substrate over here is 1 substrate to 10 inhibitor, what's going to be the likelihood the enzyme is going to bind the inhibitor? It's 10 to 1, right? So I expect I'm going to see a lower velocity because I'm going to have fewer enzymes catalyzing the reaction when that ratio is low. Up here, I might have 1,000 to 1 of enzyme to inhibitor, of substrate to inhibitor. And so the likelihood that the enzyme will encounter any inhibitor is 1/10 of 1%. So in essence, if I have a competitive inhibitor, I have lower values until I get up here. And when I get to very high concentrations, these 2 essentially become the same line. That's why when I have a competitive inhibitor, the Vmax does not vary compared to the uninhibited, because I can outcompete it with if I put enough substrate in there. If I take this same plot, however, and I look and say, well, if I have the same Vmax, I must have the same Vmax over 2. What do? Well, I plot over here, and I discover there is where this comes down, and there is the Km value that I have. And as a little side note, a couple of students have been confused about this. Why does the Km change? It doesn't really change. It's the apparent Km that changes. But nonetheless, it goes up. The Km goes up when I have competitive inhibition. Now, if I have non-competitive inhibition, I have a very different phenomenon. The ratio of inhibitor to substrate doesn't matter at all. They're not competing for the same site. All that the inhibitor does, it looks for enzyme and it binds to it. So let's say I have enough inhibitor present that I knocked out 50% of my enzyme. 50% of my enzyme. Okay? That's true if I have a low amount of substrate. It's true if I have an infinite amount of substrate. The same percentage of enzyme is going to be knocked out. if I did my experiment, my first experiment without any inhibitor at all, but I only used 50% as much enzyme, what would happen to Vmax? It'd be half, because Vmax depends on how much enzyme I use. So I know if I use enough inhibitor to knock out 50% of my enzyme, my Vmax has got to drop, because I only have 50% of the enzyme active. So in that case, my plot would look something like this. Okay? Where now, let me label these. This is competitive. This is non-competitive. Okay? And that's where I would be. Now you say, well, what happens to Km? It turns out Km is the same. And if I drew that good, maybe we'll see that the two line up. Let's see. Here is my Vmax for the non-competitive, because I said it was 50% in this case. I go over. This now is Vmax over 2 for my non-competitive. And if I go over and down, my two Kms are the same. I sort of drew that so that would look that way. But the point is that the Km does not change when we have non-competitive inhibition. Now, that's what it would look like if I did a V versus S plot. What does it look like if I do a Lineweaver-Burke plot? You could do this without any numbers whatsoever. Okay? If I told you that competitive inhibition, the Vmax stayed the same and the Km increased, well, I know that it's the same as the uninhibited, that they've got to cross at that point because that's 1 over Vmax and the Vmax is the same. And if I know that the Km increases, then minus 1 over Km must get somewhere closer to 0. I could, for the sake of this argument, pick this point and say, there it is, minus 1 over Km for the competitive inhibitor. And I've got 2 points. 2 points define a line, folks. There's my line. That's competitive. What about non-competitive? Well, if I have non-competitive, the Kms are the same. So I know that the Kms have to line up with this point, with the uninhibited, and I know that the Vmax goes down. If the Vmax goes down, 1 over Vmax must go up. So I have something like this, and there's the non-competitive. Bang. Now, I didn't use any math to do that. I didn't have to take any points and plot those, or any bunch of figures to do that. I could sketch these out just doing this logic argument that I went through here with you. You didn't see a single number that I threw your direction. You didn't see a single calculator or anything with that. These plots tell us very, very valuable things. Make sense? Everybody looks like they're stunned. Was it that good or was it that bad? Okay. Now I've got green and yellow chalk. And you know when you're wearing black pants? I hate chalk because I always go, and it looks like you're sort of, well, I won't say what it looks like, but it's nasty. You just have this stuff all over your hands, all over your pants. So, all right. Other questions? Yes?
Will you go over chemical cleavage and enzymatic cleavage?
Chemical cleavage and enzymatic cleavage, I'm assuming you're talking about a protein? Okay. So when I talked about reagents that we can use to study proteins, I talked about a group of proteases, and I talked about a chemical agent that would break a protein as well. Why are we doing these? Well, it turns out that breaking proteins at specific places is very valuable for us because when we want to do some of the analyses that we do, like let's say Maldi-Toff or something like that, what happens is the full intact protein is usually too big for us to do the analysis that we want. So we need to break the thing into smaller chunks because the smaller chunks are easier to analyze. That's why we do these protease treatments and chemical treatments to make them smaller. Well, the ones that I've talked about in class basically do, I'll just write a couple of them out. Do our work for us in terms of making those smaller pieces. So let's imagine for the moment that I had a polypeptide that had a structure like this, let's say, I don't know, glycine and alanine, okay? And I wanted to cleave it. Well, the basic thing that happens, again, we have the amino end on the left, and we have the carboxyl end on the right. All of the reagents that I gave you work on the carboxyl side of the amino acid, with the exception of carboxypeptidase A, which actually has to cut there because it cuts the terminal amino acid. Anyway, so cyanogen bromide, which we abbreviate as CNBR. Cyanogen bromide is the only one that I talked about that's a chemical cleavage agent. And the chemical cleavage agent cyanogen bromide cuts on the carboxyl side of methionine. And that's what would happen if I cut that on the carboxyl side of methionine. What that would do now is make leucine, serine, methionine, and now I'd have a carboxyl because I recreate the carboxyl. Okay? And then over here I would have lysine, glycine, alanine, and I would recreate the amine right here. If I were to treat this with trypsin, I would cut it on either side of lysine or arginine. I've only put a lysine in here, right here. And that's what would happen if I cut it with trypsin. Make sense? So there's a table. I've told you which ones in there that you're responsible for knowing. And I kept them simple. I didn't make you know chymotrypsin, for example. So look at that table and make sure you understand those. Okay? Yes, Elliot.
So with the Molotov, it talked about spectroscopy and then break these peptide bonds and shoot them out with these readings. And so what I was confused about though is how that specifically allows you to determine the amino acid structure, or
sequence.
Yeah, sequence.
Okay.
Because wouldn't you just have the light ones coming out first? And so I just didn't know how that would be.
Okay. So his question is, and he's confused about the analysis in MALDI-TOF relating to being able to determine the sequence of a polypeptide chain. So it is complicated, and it does take a computer program to do it. And what you get when you do a MALDI-TOF is a spectrum. You get a spectrum of all of the sizes of molecules that are in there. So let me just give you an example. Let's say that I had this guy up here on the top. And I don't know the molecular weights of these, so I'm going to make some up. Let's say the molecular weight of this guy is 130. The molecular weight of this guy is 140. And this is 150. And this is 160. And this is 80. And this is 90, just for the sake of argument. Okay? When I take this and I put it into an instrument, and this is always important to keep in mind, I'm not putting one of these in here. There are billions that are going in there. And they're fracturing at random places. Will they fracture at places besides peptide bonds? Yes, they will. For simplicity, we're looking at peptide bonds. Okay? But let's say that I fracture this guy, and I have billions of them in there. Some of them are going to fracture right here. Some of them are going to fracture right here, right? Some of them are going to fracture right here, some right here, and some right here. And each one of those is going to give unique sizes. So I look for the biggest thing that I can find, and the biggest thing I can find is going to be the sum of all these, right? And I start saying, well, do I see something? And I see, here's the biggest. What's the next size that I see? Well, I see the biggest minus 90. What has a molecular weight of 90? Oh, then the terminal one must be alanine. I lose 70. You start seeing? Okay. So it's important to remember that, because when we think about this, we think about one going in, but that's not what's there. There are billions of them. Okay. So we get representation of all the different sizes, and I'm simplifying for you. The computer has a very difficult task in doing that, and in some cases it can't tell you exactly, all right? Just because there may be overlapping ones and problems associated with that. But that's the basic idea of MALDI-TOF. Make sense? Other questions? Yes, sir.
Just for curiosity's sake, what is the actual type of matrix used in the multi-top?
They use different ones, but the one that I'm most familiar with is they use a formic acid mixture that forms a crystal readily and is relatively non-volatile in the vacuum chamber of the mass spec. But I don't know much more than that. I'm not a mass spec person, but what they want is something that is non-volatile in the environment of the vacuum, because you don't want it leaching things out. And you want it to volatilize quickly when a laser hits it, because that's when the starting point is for the time. And I think it's a formate solution that they use, the formate crystal that forms that they use. And it has both of those properties, works very well. Yes, Connie?
For isoelectric focusing, it's very important to have a pH gradient.
For isoelectric focusing, you create a pH gradient. That's correct.
How do you do so?
By the way, I said, so the question is, how do you make a pH gradient in isoelectric focusing? If you take and you have molecules that have a wide range of negative and positive charges, they create a pH gradient.
Oh, okay.
Because they have varying degrees of holding on to or giving up protons. Right?
Okay.
Right? So you're creating a pH gradient just by running that electrical current through it. Okay? Very good questions. I will tell you, I'm not exaggerating. I'm impressed with the questions, and this makes me feel very good that maybe you guys are really thinking very hard about this exam. Maybe we'll see a very high average, or at least among the people in this room anyway, huh? Yeah, right. Go us, right? Other questions? Connie?
Can you explain NMR again?
Can I explain NMR?
Yeah.
Well, I can explain it the way that I explained it before, which is that nuclear magnetic resonance arises from the fact that certain nuclei have spins, and those spins can be manipulated by electromagnetic radiation. The energy it takes to convert a given spin from one state to another state is a characteristic of not only the nucleus that has it, but the electronic environment in which it resides. Okay? So something that is a proton, for example, on a methyl group is going to have a different energy to change its spin than a proton that's on a hydroxyl group. Now, fortunately, we know the characteristics of those. So when we get a nuclear magnetic resonance spectrum, we see a certain energy that it takes to make that change for a given proton. We say, oh, well, that's what it takes for a hydroxyl. We know we have a hydroxyl in our molecule. Okay? What if the hydroxyl is interacting with another, let's say, carboxyl group over here? Okay? Then we have examples of one group affecting a separate group. All right? And that involves that technique I called two-dimensional NMR. It's considerably more complicated than we're going to go into here. In fact, NMR itself is more than we're going to go into here. But the value of NMR, for our purposes, is that NMR is a spectroscopic technique that tells us structural information in an aqueous solution. Crystallography, which is the other primary technique we use to do that, does not work in an aqueous solution. It requires the making of crystals, that is the precipitation of the molecules that we're interested in, or the proteins that we're interested in. So the beauty of NMR is the ability to do that analysis in a solution, very much like the environment that the protein is in the context of a cell. Okay? And I don't want to go any deeper than that, just because it's more than I've talked about in class. Okay? Yes, sir?
Are there contradictions to using NMR with the polar side chains, since NMR is the basis of like MRI imaging, correct?
Mm-hmm.
And that requires a strong permanent magnetic field.
Mm-hmm.
And it seems like if you use that with polar side groups, you'd have some problems.
If you use it with polar side groups, you'd have problems.
Because you'd have the magnetic field would affect them adversely, maybe pull them or distort the molecule in some way.
I don't know the answer to that question. His question has to do with, does NMR affect molecules that have polar side groups? As far as I know, the answer is no, but I'm not an NMR expert. I can't tell you that definitively. One of the things you see with very polar side groups, like a carboxyl, is you get proton exchange. So remember we were talking about ionization, so protons can come off and protons can come on. And so you will see considerable changes, and this is one of the ways that NMR can be very beneficial to tell you structural changes as you change the pH. And so as you change the pH of that solution, you'll see the NMR profile change because the protein structure itself is changing. But I can't tell you that there's not some induced change caused by the magnetic field. I don't think so, but I honestly don't know that. Yes, Elizabeth. I started to say Samantha. I don't know why.
In affinity chromatography, how do the proteins attach onto the group, say it's ATP or something, covalently?
Okay, so her question is, in affinity chromatography, is the binding between the protein and the group that it's attaching to? Remember that we do affinity chromatography by taking beads and attaching molecules to it that we know the protein attaches to. So the example I gave in class was ATP. So if I have a bead with ATP on it and I put protein binding molecules on it, do they covalently attach to that ATP? And the answer is no, they don't. So that is not a covalent. And the way we would wash that off is we would just add ATP. So when it comes off, it would grab the loose ATP instead of staying stuck to the bead, and the protein would wash off. Yes?
But that ATP is covalently bonded to the bead.
The ATP is covalently bonded, absolutely, because I don't want that ATP flying off.
Yeah.
Right. The ATP is covalently bonded to the bead, but the protein is not covalently bonded to the ATP. Yes, sir?
Can we do a short review of the Bohr effect?
The Bohr effect? Okay, well, that was sort of what I put up there on that plot, right? That was the plot where I changed the pH. And when I lower the pH, the protons come off. I'm sorry, not the protons. When I increase the protons, the oxygen comes off. And when I increase the carbon dioxide, the oxygen comes off even further. So that's basically what the Bohr effect is. So
on the homework, there was one that shifted it the other direction, as I recall. Was that the denaturing?
I'd have to see that. I don't know the problem you're talking about. But let's say I told you I increased the pH. What would you say would happen to the oxygen affinity of hemoglobin? Go up, right? If lowering it makes it go down, then raising it's going to make it go up. Okay. Yes?
That's a good question, but we don't need to know how to calculate delta G's or T.
You won't do any calculations on delta G, but I can promise you on the second exam you will. That makes you feel better. Does that make you feel better? Delta G, I will tell you, people after they've done Henderson-Hasselbalch, I rarely ever get a complaint about delta G problems. Rarely ever get a complaint. People find that they've sort of figured out in their head the Henderson-Hasselbalch, and the delta G problems are very, very similar in nature. So delta G, I don't think you'll see any sweat about problems on the second exam compared to what you might have for Henderson-Hasselbalch on the first exam. Yes?
When you say raising the pH will increase oxygen availability.
Yes.
And I believe there's a natural pH, and when you lower it, it will reduce the oxygen availability. How about if you raise it above the natural pH?
Okay. So his question is basically that I said when hemoglobin is in a higher pH, it'll have higher affinity, and when it's in a lower pH, it'll have lower affinity for oxygen. And his question basically is, well, what's the limits? All right. As you can imagine, there will be places where if we raise it to a high enough pH, we're going to denature the protein. And I think that's what he was asking up here as well. If you raise it to a high enough pH, you will denature a protein. So these are...
Even if you are not denaturing the protein, raising the pH above the natural pH will have some uncertain effect on the structure of the hemoglobin.
Okay. So keep in mind, these are relative things. They're relative things. So if I say that what you're calling the natural pH is, say, the pH of blood, if I went from 7.4 to 7.5, would I see a higher affinity for oxygen of hemoglobin? The answer is almost certainly I would, yes. Okay. Is there a limit to that? Yes, there is a limit to that. So that's, again, because there are relative issues here. And so the more we start to denature the protein, the less ability to bind and the less functional sort of things we're going to see with this. But in that range of the physiological pH, lowering it will favor release, raising it will favor holding. Okay? But it's a good thought. I mean, you have to think about this. There's the experiment that they did. I love the description of this experiment. Okay, so the scientist has this idea that fruit flies behave as an ideal gas. Have you heard this experiment? Okay, so the idea, the hypothesis is fruit flies behave as an ideal gas because ideal gas, we think of the molecules moving randomly, and we think about the fact that the molecules speed up as we increase the temperature of a gas, right? You increase that's basically what temperature is more impact of the gases and so forth that's happening. So he does the experiment and he writes down, at 4 degrees above 4 degrees centigrade, that is maybe 40 degrees Fahrenheit, fruit flies, move in chamber, very slowly. They're cold-blooded organisms, right? You raise the temperature up to 10 degrees centigrade and they speed up, and they're still random as heck, flying in the container. You go to 15, okay? You get them up to 37, and they're just going like this, but you get them up to about 50, and all of a sudden they cease to be an ideal gas. So the protein denaturing is the same way, right? That you take the pH high enough, it's not going to function as that protein anymore. So, yes?
Would it be a good idea to study up on purification tables?
Would it be a good idea to study up on purification tables? I think it would be a good idea to study anything I've talked about. So I'm not going to, but I can't say yes, this, no, this, etc. Okay, because your question is this going to be on the exam? I think I'm paraphrasing, but the point is that anything I've talked about on the exam in class is fair game. Okay, anything I've talked about is fair game. Use the highlights as your guide. Yes.
So that means that just if it appears only in the textbook reading, it's not fair game.
If I haven't talked about it, then I'm not going to ask you. I've already promised you that. I'm not going to do that. Yes.
Can you describe the perfect enzyme and what that means?
Can I describe the perfect enzyme and what that means? Yes. Well, as a class, you guys sort of agreed that the perfect enzyme would be one that has a very high kcat and has a very low km, because it would mean it would take very little substrate to get it to half of Vmax, and once we got it to Vmax, it would be going through the roof. So that's really what is an ideal enzyme. A perfect enzyme is one that the ratio of kcat over km is very high, and it gets to a limit. And when it reaches that limit, we call the enzyme perfect because it no longer can be improved by mutation. In fact, every mutation we try to induce will lower that ratio. So it has evolved to its maximum efficiency, as it were, and it's working really, really rapidly. That would be the definition of a perfect enzyme. A perfect enzyme is perfect, and it can't get any better because the substrate can't diffuse through water any faster. There's a limit. And so the enzyme is sitting there waiting on substrate. It can't go any faster because of that limit. And that's what a perfect enzyme is. Yes?
So to clarify, when you say dissolve through water, it's like it can't reach the enzyme just faster.
It can't reach it fast enough. So if we think about carbonic anhydrase, I said we can make 1 million molecules of product per molecule of enzyme per second. We start thinking that there's 1 million of those carbon dioxides that are hitting one enzyme active site in 1 second. There's got to be a limit. There's got to be a limit at how fast a molecule can diffuse through water. Brownian motion essentially defines what that limit is. And the medium of the water itself, being viscous, is also a limit. So that's what the problem is, why we can't get beyond a certain point. I mean, logistically, 1 million a second is pretty hard for me to conceive of in the first place. I'd say 20, 30, 1 million? I don't get it. That's why I say the nano world is different than the world we see. Yes, Elliot?
Are there any examples biologically of enzymes producing an environment where the medium's not water, so it's something, they produce something else that the substrate can diffuse across faster?
Well, so his question is, are there examples where it would be produced in an environment where the diffusion of water wouldn't be a factor? The only place I could think of something would be if we had, let's say, an environment of ethanol, perhaps. And I know of no cells that can live in pure ethanol. Because if we think about it, a cell if it lives in pure ethanol, which it might be able to do, won't have the ethanol on the inside of it. On the inside of it, every cell is aqueous. So even if we could find things that cells could live in, it still doesn't affect the overall thing. There are no cells, 0 cells, that have inner environments of anything but water. And that's where the enzymes are. So the answer is basically no. There are speculations that, I'm sure you guys know this, of the moon Titan that orbits Saturn. Titan is a phenomenal place. It is incredible because it has probably more carbohydrates on it than the Earth does. And carbohydrates, of course, are building blocks for more complex molecules. It rains ethane and it rains methane on Titan. Those probably have very different viscosities and coefficients of viscosity that are there. So if there's life on Titan, perhaps they have better or more perfect enzymes. I don't know. That's the one that comes to my mind. Yes?
So I have kcat equals maximum velocity over concentration. And
Concentration of enzyme.
Enzyme. Okay, that was my question.
Yep. So kcat is equal to Vmax divided by the concentration of the enzyme. Not just concentration, but concentration of enzyme. Yes, sir?
We consider the oxygenation and deoxygenation of hemoglobin in terms of denaturing.
Do we consider the oxygenation, deoxygenation of hemoglobin to be some sort of denaturing? And the answer is no. No.
And you're changing the shape.
You're changing shape, but denaturation implies loss of function. So I would say no. I mean, these are semantic arguments, but basically that is not a denaturation, no. But you're right, shape does change. And we're going to see a lot of shape changes. I'm going to talk about the mechanism of action of an enzyme called ATCase. I think it's next week. It could be the week after. I can't recall. And ATCase is a really cool enzyme. It's got 12 subunits. Okay? 6 identical catalytic subunits and 6 identical regulatory subunits. And there's some big shape changes that happen there. But the enzyme is functional, irrespective of those shape changes. So when we talk about denaturation, we're basically talking about a shape change that's a loss of function. It can't go back. Okay? Okay. Have we exhausted the questions? All right. For you guys and also for the people in TV land, I will give you my cell phone number. If you have questions, you are welcome to call me. I meant to give it in class on Friday. 541-908-4465. And for the people in TV land, I'll even write it on the board. 541-908-4465. That is me. Yes, ma'am.
Sorry, quick question. Do we need to know the examples that you gave in class?
I don't know what that means. If I've talked about anything in class, you are responsible for it. If it came out of my mouth,
Okay.
you are responsible for it.
Okay.
Okay. Okay, you all ready? You'll be ready.
Close enough for government work. Thank you.
You bet. Study hard.
There's no other way.
That's right.
You're not studying.
What
I had one question about when you were talking about the ideal enzyme and the perfect enzyme.
Yes.
It was a very high K.
Very high Kcat divided by Km.
By Km, so Kcat.
Kcat to Km ratio is very high.
Okay, and so with a perfect enzyme, it's the highest that it could be.
With the perfect, that's right. That ratio reaches a maximum and it doesn't go any higher.
Okay.
Yep.
Okay. Thanks so
Man,
much.
you have my sympathy transcribing me. I had, Joe Alexander probably.
Yeah, she's my supervisor.
I'm sure she's told you the story about she used to sign my class.
Oh yeah.
And I talk fast. When I get going, I can just blah. And they're technical. She had to spell out names of enzymes. And stuff. I always told her, I said, "You're a remarkable woman." That's all I could say. I mean, absolutely remarkable.